Well, let's freaking go then. Let's freaking go. Oh lord. Hello. Hello everyone. Today we will maybe need to change with won't be turning the whole time. Okay, no problem, no problem, no problem. It will be very basic. You'll see. You'll see. What is this music called? You mean the one for the intro? Do you mean the one for the intro? Because the one from the intro is from Punishing Grey Raven. Let me see how it's called. It is Punishing Grey Raven. No, or just Grey Raven. Or Grey with an A. Yeah, it's Last Resort. Fit Kinoko. It's Last Resort with Kinoko. I hope that everyone had a great day. I was in a bit of a rush because I overslept, so correcting and making more of my slides was a challenge. Let's say that it was a challenge. Won't. Oh. Still want rendering. I'm using my famous. I kinda want. I would love to make some rendering. If you have a recipe, whenever I find some beef, oh, I would love to. Because of the way you described it, mwah, absolutely delicious, beautiful, and nutritious. No, this isn't blasphemous, unfortunately. This is education. You are now trapped with me. I'm an education tuber now. I'm an education tuber. Seriously, what's this track called? Yeah. No. It's seriously Last Resort, Fit Kinoko, Punishing Grey Raven. If you're talking about this one that is playing right now, it is called Rex Lambo. Alone by Rex Lambo. Let me just make a parameter for this one. Yes. I just want to. No. Property. Is the most and well, that will be a very basic look at what biology is. And basically I'm going to give you the very basis. Let's go with that. Let me just adjust the thing. I just want to adjust the thingies. Okay, maybe like that. I want to have a full screen. I want to have a full screen included there. I should have made it earlier, but you know how lazy I am. Reject meat. No. I am made of meat. I am a parasite Vtuber. Design, draw, animation, slideshow. Yes, from the beginning. Freaking hell. Okay, so I'm going to be seriously. Okay. So it's going to be like that, huh? It's going to be like that. Why not? Why the fuck not? Okay. Let's begin. Why you ask me? Why you ask him instead of me? Let's do some parasite classes. Let's be. Let me just. Another one. I just want to. Yeah, I just want this media source. If it's possible, source media 5. Okay. Because I still want to have music. I still want to have some sort of music, some sort of ambience. So let's go with that. And let's begin. Oh boy, I don't know if I'm excited or if I'm going to die from stress. Because I am extremely nervous and I want this thing to actually work. I want this thing to actually make you learn things. You will read and you will learn. Yeah, let's go. Let's go. And welcome to our little class. Welcome to our little class here. Because of a delay, I am not going to be able to really be able to interact way too much with YouTube. Twitch is way better when it comes to a delay. But I will answer your questions. Since we are a very small group, we can make it as interactive as you want. I will ask if there's any question. I will wait about like 10 seconds. So it's going to be 10 very awkward seconds of silence and just maybe me rambling about. If you have a question about any slides, do not worry. I will absolutely go back if necessary. Also, if you want the slideshow, if I find a way to make it anonymous, I will absolutely give it. Or maybe you can just, copy the thing. It will be on YouTube. It's going to be one of the VODs, so it's going to be all right. It's going to be all right. I'm just going to take my water. My water is here. And let's begin with the Parasite class. So, what we will be learning in this episode is going to be a very quick intro into biology, into what I would say the very fundamentals, which is looking at the question of life. What doth life? Very important. One of the main questions of all the philosophers and the searchers of truth out there. The criteria for what can constitute life. Very simple for Random is Ask Google. Oh, okay. No problem, Reinhardt. A quick recap on what abiogenesis is, and no problem. Do not panic, I'm going to explain it to you. The 4 ingredients of life, plus the fuel, and then the 3 great domains. That will be very, very succinct. I am not going to go into the details because we could be here for days and I only had a few hours to make this slideshow. I don't intend on making this more than 2 hours. I hope, I don't know. You have actually forgot to make it. Let's go. Let's fucking go. First slide is looking at the question of life. Okay. So, why do we look at the question of life? Oh shit, that's not good. This one is not good. That was post-modification. Yeah, do not make your slideshow on the rush. Do not rush your slideshows, or there's going to be a lot of exciting problems that are going to come with it. But yeah, it's about finding life in unexpected places. So, looking at the question of life allows us to basically make it more abstract and liberate ourselves from what I would call preconceived notions of what you would find in life. Let's remember that for the longest time and for most people, insects are not animals and plants are not considered alive and microbes do not even enter into the context. It's very confused. And for example, there has been a finding of aerobic, so microbes that do breathe air or oxygen rather. Microbial life persists in oxic marine sediment as old as 101.5 million years. So think about it. You had microbes that were trapped in rocks for 100 million years. The rocks contained oxygen, so the microbes had no choice but to adapt and evolve into extracting the oxygen from the rocks to breathe. and then reproduce from there. They have a very, very slow metabolism because of a very, let's say, they don't have a lot of nutrition out there, but it's still there. They can still derive energy from inside rocks that were caught inside the Earth's crust. So life is basically everywhere. In the air that you breathe, in the fucking in the sky. It's YouTube. I don't have the right to say a lot of curse words. In the sky, in the earth, literally in the earth, and also inside of you. So there's also this thing. It's very important because if we were to look at astrobiology and finding new life, we need to have some criterias of what we would consider life, rather than what is not life, because there's some chemical reactions that are very interesting that can happen that may be confused for life. For example, a mercury plus any aluminum, it makes a very fast growing structure that can be considered a growth and chemically complex. Still not life though. Still not life. There's also a big problem with this question, and it's about asking the right question. The right question. What would be the right question? Well, what is life is just like what is love. It's something that is applicable for alchemy, traditions. It's more philosophical than practical. It gives substance to the word life. Basically, it gives it its meaning. All the philosophical meaning that we have, and we have a little philosopher, a little Xavier, on the road to finding what doth life. I am a traveler, and I am a survivor. We are an extinct breed. We are an endangered breed. Let me go there. Xavier. Please do your thing. Do your thing elsewhere, Xavier. So what is life is unfortunately not extremely practical. For one freaking gag, Xavier is literally. You're not reading a lot. So for the purpose of this class, what is life is not the right question. So what we're going to do is create a life box. So we create a box called life and ask what can we put inside the box and separate it from the rest of the universe. And that's a very actually question. That's a very actually kind of reasoning. But don't worry about it. We all hate those kind of little guys. We all hate the little actually stuff. But it's very important for this class. It's very important. It's very important because if you want to be a good parasite, you need to know what exactly is life and what you can actually infest. So, the criteria to enter the life box on Earth, because if you were to say, well, if it's not on Earth, maybe it doesn't fit those criteria. Yeah, buddy, we actually need to search based on what we know. So, of course, we are going to base our criteria on the only form of life that we know. So, on Earth. Which is why we are searching for water. Because water is the ultimate... it's the ultimate solvent. It does a lot of very critical chemical reactions that we need to live. That are not found in any other chemicals. So is carbon, too. So, what enters in the life box? What are the criteria? Let's say that you are a bouncer in a cool place called life, and you have all those things that want to enter the box called life, and you have to be a bouncer. You have to say you are not on the list. You are not on the list. You are not wearing good shoes. Get the F out. Get the F out. Oh, YouTube. So, what you need is to be chemically complex. So, anything that is akin to a diamond, which is made of one single element, in this case carbon, it's not alive. If you want to have a reaction of life, you need to have a lot of elements, mainly carbon, nitrogen, hydrogen, oxygen, and a few other elements. But carbon, nitrogen, oxygen, hydrogen, the main ones. It's the big four. The big four. You're going to see carbon, hydrogen, oxygen, and nitrogen a lot. Because they are able to do a lot of cool reactions between themselves. What we call organic chemistry. It's made out of definable enclosed limits. It's cells. What we call cells. So, viruses, or other dead stuff. We don't want them. They're not cool. They're not going inside of a life box. Rocks either. Rocks are chemically complex. There's no cells in rock. I mean, there's no cell rock. There's no rock cell. And even if there were, then rocks are not capable of auto-replication. You do not see a rock shitting out another rock, and then the rock grows according to metabolism. So possesses its own capacity to keep stuff in and keeping stuff out, very specifically using energy. Now, if you put rain on a rock, water is going to go inside of a rock. The rock is going to be modified because of the water. It is very, very bad. Capable of auto-replication, that puts out viruses. Viruses are not alive. Sorry about the people that are believing that. Viruses are absolutely not alive. They do not fit these criteria. And these criteria in the sense that they do not have their own metabolism. Technically, they do have an enclosed limit, but it's not a cell. So get the heck out, viruses. And again, dead stuff. We are going to exclude dead stuff because technically they are chemically complex. They have cells. At the very beginning, dead stuff do have cells. And those cells do keep stuff in and keep stuff out temporarily. But they do not have a metabolism. They do not use energy. And they do not reproduce. You do not see a skeleton in a coffin meet another skeleton and then making baby skeletons. That would be horrible. Imagine your grandma doing that. She dies and then she becomes a skeleton and then she makes other smaller skeleton and technically those skeletons are related to you. No, that's horrible. Think about it. That's absolutely freaking horrible. You do not want to think about that. Also, zombies. Zombies are not alive. They do not reproduce. You do not see. I mean, I've seen that. I've seen that if you go on some websites, 2 zombies doing it, and then making another smaller zombie. Not the smaller zombie parts, but still. Oh, I did see the smaller zombie parts on some websites. Huh. Culture, my friends, is going to ruin your life. So, we are going to enter another word with a big word. Ah! Biogenesis. It's a very big word. And I know that some people are very intimidated by big words. But we use big words because big words are exact. They offer an exact definition. And that's what we're going to use. Yeah, culture. Oh, heck, culture. Such a bad thing that happened. What? How come that I don't see chat? Huh. But yeah, unfortunately, abiogenesis happened, and that's the main hypothesis on how life happened on Earth. And I say happened, not was created. In this class, we exclude the existence of a possible creator or possible plan, but it is just not the case. We are going to strictly ignore those things and think about life as a completely independent structure, because otherwise we're not going to be able to interact correctly with it. Holy shit, there's so much delay. There's so much delay, my dude. I understand now. Yeah, I understand why streamers have a lot of problems. YouTube, you have so much delay. Please help me help you, YouTube. I want to have less delay. On Twitch, I can interact nearly nearly instantaneously to comments. So please. I want to have that back and forth. Also, does anyone have any question about the definition? Any questions about the criteria of a life box? Any criteria about the any questions about the criteria of a life box or anything anything like that? Any commentary? Any insults? Any jokes? But yeah, for me it's funny that viruses are made of biological stuff, so they are completely complex, they are definable by your limits. And technically once they enter a cell, they do have some kind of metabolism that is not their own. And they are not capable of auto-replication, so viruses by that very definition are not alive. Which comes into contention. So it implies that there is a life spectrum of not alive, and then alive, and a lot of stuff in between. But let's go for definitions. Let's go for the favorite stuff. Definitions. A, biogenesis. It's all in Greek. A for non-bio for life, and genesis for origins. So origins that are not that do not include life. So the origin of life without life. Also called autogenesis, it's the hypothetical process by which living organisms first arose on Earth from non-living matter. So from thingy not alive to thingy alive. And to an obsolete name for spontaneous generation compared biogenesis. Spontaneous generation is not going to be included here. It was the prevailing theory during the 17th, a good part of the 19th century, until we had the microbial theory. And basically, spontaneous generation was that if you let stuff die, from big dead stuff, you have smaller alive stuff that emerges. From carcasses, Greeks did the hypothesis that flies came from meat. Maggots and then flies came from meat. Like meat was transformed into maggot and then flies if you let it out for long enough. Which was proven wrong in either the 16th or 17th century in Italia. And what the dude did is that he took a jar. He literally took a jar. He put a piece of meat inside. So two jars, two pieces of meat. And what he did is that in one he let the meat out in the open, so the jar was not blocked. In the other, he simply like blocked the jar. He put in one jar that was blocked a cap. In the other, a simple strand of tissue, so bandages. And well, when there was bandages and stuff, the meat rotted, for sure. The meat did rot, but no flies. And when you see the meat that was left in the open, well, you see flies, and that wasn't self-evident at the time. That simply wasn't self-evident at the time. It was an accepted idea that big dead stuff did create smaller alive stuff. That was, by definition, then inferior, because it was emerging from the collapse of a bigger system. And I think it was posited by Aristotle. It was a cool definition at the time. But let's say that science marches on. Also, this doesn't make any sense. Here, that's better. This is way better. So, where did this abiogenesis happen? Very, very, very easily, you can say that abiogenesis happened on Earth, but a different Earth. Not exactly the Earth that we know. In the very beginning of the oceans on Earth, the oceans were green, the atmosphere was orange. There was literally no oxygen to give the sky its bluish tint. there was a lot of iron in the ocean. Iron oxides. And if you know about your stuff, you know that the ions Fe2 plus. So let me write it. Fe, and then I would need to put it in. I think it's uppercase. No, that's not uppercase. That's not uppercase. I don't need. No. You know the stuff. Let's just call it Fe2 plus. Those are the ions. So iron that lost 2 electrons. The seas were literally completely saturated with that, and this is where life appeared. You have to understand that the atmosphere was also extremely different. The orange was due to methanes, to some species of nitrogens from hydrogen. It was a completely alien planet. The day was only 18 hours. I think it was only 18 hours. I think it was 18 hours a day, one day, for a rotation of a planet. And every day, all the time, the moon is basically slowing down the Earth. So in about 4 billion years, or however much has happened since the moon appeared, the Earth has slowed down its rotation by 6 hours in a few billion years, because of the influence of the moon. It's pretty cool, right? So you would go into a time machine and go back very, very, very far away in the past. The Earth is completely different. Absolutely impossible to recognize. So there was a guy, Muller and Urey, 2 guys, Muller and Urey, that had this experiment, and they said, okay. So Earth was like that. We theorized that there was also a lot of meteorological events because of volcanism. So a lot of thunder, a lot of lightning, a lot of meteorite strikes, because the solar system was still very young. So there was a lot of energy in the sky. There was a lot of energy. And maybe with these very simple molecules that we have, we can, and the energy that is given, we can spontaneously make more complex molecules that will then be the building blocks of life afterwards, that are going to be found in the oceans through rain. They did their experiment. So you see a lot of water from the oceans, a lot of methane. This is I don't remember. This is ammonia. And this is dihydrogen. So a lot of reductive species. We call them reductive because they can give protons. You see that they have a lot of hydrogen. They can give a lot of protons. And this is very important because thanks to the lightning, we are going to break down those protons and give them to other species that are going to be their oxidizer. You see that the water has some stuff, but normally it's just water. It was pure water at the very start. The atmosphere was the important stuff. Then the atmosphere basically cools down after it had this lightning spark, this constant lightning spark. Goes inside the trap after it condenses. The trap allows us to have a sample. And then the heating source, because of a heating source, it goes back inside the oceans. And thanks to a heating source, we go back there. There's going to be another time for boiling water. And it goes back. It's a cycle. Again and again and again and again. And he did that for a few weeks, I think. And in a few weeks, he found sugars. He found amino acids. He found nucleic acids. Very simple ones. But you have to understand that it was a huge discovery. The spontaneous creation of organic molecules from nothing but energy and the component species. This is what inspires abiogenesis. This is for a moment the leading theory. And another thing that is very important. We have found organic compounds in space. I know that you know. I know that everyone knows that the center of the galaxy is supposed to taste like strawberry. Or was it? No, it's raspberry. I think raspberry. And it's because of those very complex compounds that basically are there through nothing but water, themselves, and energy. The gas phase process, ionization, dissociation, and two-body reactions. And through there, you can find very complex, very complex molecules. Understand that it's not self-evident. We had to do this discovery to accept it. We had to do it. And, well, because of that, we have accepted that there was an abiogenesis. That with these very complex molecules that were formed, they arranged into what we call protocells. And let me give it to you. Let's go. You have early Earth molecules. You have water, carbon dioxide, ammonia, methane, dihydrogen sulfide, so sulfur. Here you have cyanide. This is glycine. I think that this is glycine. 2, 3. Yes, this is. Yes, this is glycine, and this is CHO2. I think that this is methanol. I'm not too sure anymore. But you have clay, and clays are very important because clays basically give you this stuff where you have a lot of electron exchange and minerals. Through that, you can have the first reactions. The very first reactions with prebiotic chemistry that creates lipids, some complex nucleotides. We are going to see them. Some complex amino acids. And after that, it arranges itself because it's the most conservative, it's the most energy conservative element. So you have the first self-assemblies thanks to the clays. Because the clay is just there, you have a molecule that goes inside the clay, they just arrange themselves. And after that, it becomes very self-efficient. After a while, you're going to have these molecules that goes inside these vesicles. The vesicle is just a bubble of water surrounded by fats. And this fat is special. It's not just fat. Fat is very special because it has a polar head and an apolar water theory. I'm going to call it a water hydrophile and hydrophobe. So it has a hydrophilic head and a hydrophobic tail. So the head is going to like water, it's going to go towards water. The tail is going to arrange itself so it doesn't go towards water. And spontaneously, it's going to arrange itself so that you have water outside, water inside. The heads protect the tails from the water. And you have this bubble. So this bubble is a closed environment. This is the first operation on what you could call a cell. And a cell ultimately is just that. It's a bubble of water protected by fat with some structures inside of them that can self-replicate and do all kind of very wonderful stuff. And has its own memories too. And can grow. So this fat can flip. So this can go there. And they flip inside, giving water inside, making water go inside of the arrangement. Yes, but also stuff that is inside the water. So you can have some very, very, very simple pre-arrangements that are then going to be a bit more complex And then you have the earliest life, the earliest thing that you can absolutely call alive. So as you can see, life is very much a spectrum. It's very much a spectrum. This is not quite alive, but it's there. You know, it's right there. And then you have the first thing that you can really call life. That is going to be pre-assembled, that is going to be reproducing, that is going to be able to have its own metabolism. Very cool, very good. Where did it happen more specifically? Because Earth is a bit vast, you know. Earth is a bit vast. It's a bit vast definition. It must have happened in things that we call white smokers. These white smokers are basically just thermal vents. They are not black smokers, because black smokers are a bit more violent, just a bit more violent. But these white smokers allow ourselves to have a lot of energy-rich molecules that come from the inside of the Earth, that go inside the water. This is just what you see here is not smoke. We call it a white smoker because it looks like smoke. But it's just water, superheated water, with a lot of mineral molecules that refuses to mix with the water that surrounds it. But as it cools down, it's going to mix, and it's going to fall down. And you see here the yellow stuff here, it's sulfur. It's mineral sulfur. The gray stuff here can be some kind of iron, because as iron binds with the oxygen contained in the water, well, it's going to become rust, what you would call rust, mineral, let's say well, iron oxides that are going to be able to go to a solid state, not mix with the water, and go to the bottom of the ocean, where it can be exploited by life. So this looks a bit desolate for us, but you have to understand that this is a gourmet 3 meals assured a day state for all kind of life that we call chemotrophs. We are going to go to trophies next week, we are going to go to the way that life actually eats next week, because it's pretty interesting. But you have to understand that those things derive energy from the high energy molecules that come from the inside of the Earth. They don't need light, they don't need oxygen. Sometimes you just need high energy molecules. And that was the case for most of the history of life, until we had the way to breathe down, to make oxygen, breathe down oxygen, and even after that, it's a minority of the history of life, maybe. But oxygen was a freaking game changer. It allows us to have so much more energy, to be so much more competitive, and boosted evolution in a way that you cannot comprehend yet. And so the question is, given that all the conditions here are basically able to give us new kinds of life, why don't we see another kind of life that appears? Well, the problem is basically exactly the same problem as me as a new VTuber on a platform, compared to other very big VTubers that are here for years, or Hololive, or FaceConnect, or Niji Sanji. We get eaten up. We basically get stuck at 5 views. We get eaten up by the rest, and after a while, some of us, well, we abandon. And that's exactly the same with the new life. What I'm going to call this new life. New life is not going to be prepared, because look at that. It's super simple. It's absolutely freaking super simple. This is basically a gourmet chef. Yes, Oxygen was a competitive matchmaking update. Absolutely. So this little thing, absolutely inoffensive, is going to be a freaking gourmet chef compared to a modern bacteria that is here with toxins, with a way to make poisons for other bacteria, to communicate with other bacteria, exchange genes, but it has its own protections. its own way to protect itself, it's just going to see that it's going to say, very appetizing. Let's go, let's eat it. So yeah, if new life appears on Earth, and I'm convinced that literally every year there's a lot of prebiotic state of quote-unquote new life that appears, well, it's going to be eaten up almost instantly by the competition, which is sad but true. You have one second of evolution compared to 3.5 billion years of the most competitive game ever. So every single player out there, humans, animals, plants, bacteria, archaeobacteria, we are also competitive, and we are always competing between ourselves. There's no new place for new life, because new life has to have at least 1 billion years before it becomes a bit competitive for itself. It's the perpetual. It's a new player. Imagine you put a very new player of League of Legends, Apex, even Fortnite into a ranked match with world champions. Yeah, that's exactly what's going to happen. They're going to be bodied instantly. They have no chance. Absolutely no chance. So maybe it's there. Maybe we can create it. Maybe we can make it. Maybe we can see it. But it's never going to be relevant. Just like me. And now that we've seen that, we can go and see the 4 ingredients of life. And begin with the most well-known. Nucleic acids. Which allow us to have memory and energy. Through the arrangement of 4 little new bases, you have programming. Life invented programming before the humans did, before our brain did, which is pretty cool. But instead of ones and zeros, we have A and G, and A and T, C and G. We read A, C, G, T, etc., etc., etc. We translate that into some other stuff. You're going to see there's a logic there. There's a logic in the way that I categorized it. And in DNA, something interesting. A has to bind to T, and C has to bind to G. And the reverse is very true. C has to bind to G, and T has to bind to A. So you just have to read one strand, and you know the anti-strand. You know the anti-strand. It sounds so cool. It sounds absolutely so cool when I say anti-strand. Rather than, you know, just the freaking loser nerd shit that I'm explaining right now. You also have DNA is what I would call long-term memory. Well, RNA is very cool because it can be used in order to be a machine. It can do some stuff. It assembles all the proteins in the form of ribosomes. It's also the way that this thing is expressed and translated. So RNA reads RNA in order to make proteins. Yeah. This gets translated into RNA to be read by RNA in order to make proteins, or maybe sometimes just arrange itself into a thing that will read into a protein. It's absolutely insane stuff, and this is what we call translation, because you need to translate DNA into RNA, and RNA is pretty much made of the same stuff. It's guanine, it's adenine, so it's G, A, C, and U. Also, it doesn't bind, so you don't have the pairs that we've seen before. Or does it? It does bind sometime. Sometimes it does bind within itself. And with RNA, all the rules are off. G can bind to A sometimes, G can bind to U sometimes, C can bind to A sometimes, and the G can bind to G too. Sometimes. And there's also non-canonical bases that do exist, like xanthine. What else? The only one I know is xanthine. Something that is very cool is that you can buy xanthine and caffeine and you would be able to make your own life form with its own code, with its own way to express RNA. It's very cool stuff. It's very, very, very cool stuff. What is uracil? Well, uracil, as you can see, is just thymine without this little group here, without this methyl group. So you get thymine, you cut out the C, and that's about it. What is not shown here on those voices is the sugar. There's always a sugar and a phosphate. As you can see here, this is adenosine triphosphate. And let me tell you something else about nucleic acid. This is the true energy of the cell. All of your metabolism, all of the stuff that you do, has to be paid with adenosine triphosphate, or sometimes adenosine diphosphate, but it's never the case. Usually, you do adenosine triphosphate to break it. It makes adenosine diphosphate and an inorganic phosphate, or adenosine monophosphate. So you cut it right there, and you have this diphosphate here that can sometimes be used in some reactions. But this is the V-buck. This is the big buck of life. Every single form of life that we know, every single one of them, has to use that. This is their main energy source. And when I mean energy source, this is the jet fuel of life. It is super energy dense. Because this binding here, we're going to do a bit of quote-unquote chemistry. This link here in the molecule, super ultra energy dense. By breaking it, you liberate a lot of energy. And that's how basically all of the chemical reactions in the body happen. You break something, you break one of those, and you liberate the energy. Either here or here. Usually there's more energy here because it's less stable. And all of the game of life, and what the oxygen competitive matchmaking update did, is that when you use oxygen, you make more of that, and it's easier to make it. I mean, there's a ton more chemical reactions, but it's easier to make it, and you have more. If you have more, and it's easier to make it, you're going to have way more energy, so you can do way more stuff. You can be way more complex. You can use more energy. You basically are quicker, stronger, faster, more intelligent. That's how it works. That's how it works. This is the gold. This is the V-buck of life. This is the Roblox. Oh yeah. Everything is battling just for that. We are always battling just for that. We don't really... I mean, when we die you know how we say that the mitochondria is the powerhouse of the cell? It uses sugar and oxygen in order to make that. Also, it's kind of a pump. When we will get there, you're going to see it's going to be extremely cool and good. Extremely cool. Now, amino acids. Let's talk about amino acids, also called fucking proteins. That's how you get your gains. Heck yeah. Proteins, the machines of life. Proteins can do pretty much anything. Yeah. It is structure. It is movement. It is reading information. It is making information happen. It is poison. It is medicine. Proteins are basically anything. And in the body, proteins are made with 20 base bricks that you can see here. And thanks for protein, sir. Thank you very much. How do we get our gains? Well, we have these little proteins, these little amino acids, and we combine them to make proteins. How do we combine them? We call we do what we call a peptidic chain, and the peptidic link is basically a covalent link. If you know, a covalent link is just, you know, the link that you see here. It's just a link that you see here, atom to atom. Because there's, you know, there's a hydrogen link, there's a there's hydrophobic link, etc., etc. No, this is a peptidic link. The peptidic link is basically weaker. It's weaker than a double one, than a double link, but it's stronger than a single one. And it's between one nitrogen here, the nitrogen that you find here, and the oxygen that you find here, because they are free. And proteins are just arranged by the way they're shaped. It's literally just the way they're shaped that is going to make their function happen. Some of them are extremely simple, and I had to learn this fucking table. I can cite all of them to you. I could have drawn them from memory once upon a time. And some of them are extremely simple, such as the human, like, you know, the human regulator, the proteins, or rather the peptides, when they're very small, we call them peptides, that regulate sugar inside the blood. You have glucagon and you have insulin. Insulin just says, bro, you have too much sugar. Cells need to take the sugar inside of them. And glucagon just says, bro, you don't have enough sugar in your blood. Release sugar. Or maybe that's the reverse. Let me see. Insulin roll. I think it's that. Glucose level in bloodstream. Yeah, that was it. Insulin, you have way too much sugar in your blood. You need to stock it. Glucagon is you don't have enough sugar. You need to release it. And you know, it's pretty simple. You can draw them pretty easily. So you have some wacky links here, but that's between cysteines. Cysteines are very weird. There are some exceptions here. We have something that we call non-canonical amino acids in our in the freaking genetic code, because the genetic code for that basically. We're going to get to the genetic code in another episode. Don't worry about it. Yeah, that's about it. That is just a messenger. Or we can have titin. What we call titin, which is the longest word in any human languages. The true name for this molecule is the longest word in any human languages. It has 19,000 letters, if I remember correctly. This dude is trying to recite it. It takes him 3 hours, and I think that he abandons halfway. It's a very well-known video from quite some time. So yeah, titin is extremely big. It's found inside muscles. So thanks for proteins, sir. And then you have sugar. Sugar is super cool because it provides you with structure and energy. Respiration is just how to make sugar into adenosine triphosphate, or ATP. It's just how to make sugar into ATP. That's it. Respiration is just that. And you can, or maybe it can be structure, because remember that? Remember this ribose? Oh, look at that. Look at that. We see it everywhere. Sugars are absolutely everywhere. And basically they also make up what we call cellulosis, which basically is what paper is. Paper is just sugar, but you cannot digest, unfortunately. And lactose is another thing that I cannot digest, because I don't have the enzyme that cut this link. Yay! And it's just, oh my god, look at that. It's just glucose and another one called galactose. That's a more complex sugar, and you can have more and more and more and more complex sugar. That's about it with the sugar. And we can use other things like fructose for respiration, but we need a phosphate on the fructose. And I don't know if I will do an episode on respiration more than the basis, because respiration is freaking advanced. Like, it's super freaking advanced. And it's really difficult. But yeah, sugar is pretty simple. They're there. Structure and energy. That's how you get them. So you need sugar. You absolutely do need sugar in your diet. Not too much sugar, because sugar is naturally present in everything. But you do need sugar. And then you have fats, which is another one, structure and energy. Well, protocells. Do you remember the protocells from before? Look at that. Well, here we go. Here we have them. Here we have them. That's the fats. This is a triglyceride, and this is the main way that fats are stored inside of your body. And this is cholesterol. Cholesterol, which participates in a lot of hormonal functions. So a big messenger, very important. Fats are very important. You need to eat the right kinds of fats. Otherwise, well, you're going to not be able to produce this, or have a good or something that has a good movement in that. So fats, very important. If you want to have gains, you need to have some fats. You need to have some very good fats. And that's about it. Also, fats can be broken down in order to have energy, because it can be broken down in order to have, once again, ATP. But that's about it. That's about it with fats. Fats are very complicated. If I were to go inside the metabolism of fats, you're going to see how complicated soon enough, because I'm going to talk about the difference between bacteria and archaeas in a very succinct way. And now we go inside life. Life that we know. Basically, it's pretty much all of life. This is the tree of life. We go from a last universal common ancestor that we call Luca. And this last universal common ancestor, we use a very conserved RNA. If you want to know it, it's the 16S RNA that is used in order to do a phylogenetic. So philo is a tree and family. Genetic, tree of life. That shows you how everything is linked. Absolutely everything is linked. However, this tree of life is very, very biased towards eukaryota, because you think, oh, there's not so much bacteria and archaea. You have to understand, most of the stuff that is alive, right here, are bacteria, are prokaryotes, they basically constitute about, I say, 97% of life, as far as I remember. So a lot of things are completely monocellular, and a lot of life that you know is a complete minority of life that doesn't really matter in the grand scheme of things. No, it's not that it doesn't really matter, since the Anthropocene does happen. But if you were to talk about the diversity of life, well, bacteria are a complete and absolute crushing majority. They were from the beginning, and they are going to be here in the end. And maybe after that. And since they are very adaptable, they evolve extremely fast. That's why, if we do astrobiology, we search for microbial forms of life, and we do not really hope for animals or plants, etc., etc. Because you have to know that life spent 2 million years, at least, of its existence. No, 2 billion years of its existence, without once assembling one cell with another. And bacteria can be multicellular. They can have multicellular arrangements. And then you have archaeas. Pretty cool, huh? And this is bacteria too. This is archaeas. And I think that eukaryota here are going to be kind of these colors. These colors right there. This is us. Hello us. We constitute absolutely nothing. We constitute absolutely nothing here. We are nothingness. compared to archaeoglobi, bacteria, etc., etc. So, sometimes people thought at the very beginning that archaeas were pretty rare, and they're mostly extremophiles, But you have to understand that the very beginning of life was there, under extreme pressure, in temperatures that you would think are not extremely good for life, for yourself. Like 200 degrees water doesn't seem very good for you. Like superheated water, not really good. Well, you have to understand that for us, we live in absolutely frigid conditions for life that lives near these thermal vents, black smokers and white smokers. So it's completely relative. So is the fact that cryophiles. We're going to do the phillies in another episode, maybe pre-recorded. I don't know. I don't think that they will be pre-recorded. So yeah, crushing majority of life, bacterias. Now we go with prokaryota, and prokaryota, well, bacteria. They have a lot of anatomical discrepancy, of anatomical, how to say, diversity, but they are relatively ignored because they're not like us, they're everywhere, so people don't really care, unless you are a microbiologist. In that case, you're Chad, sir, and or madam, you're an absolute Chad. You see that they have what you would consider, quote-unquote, a pretty simplistic arrangement. But it's basically what you don't see anatomically as complex. When you say majority, are we taking numbers of individual organisms or pure biomass? Yes. Individual organism, number of species, and I think biomass too. And because viruses are even more, there's even more viruses than bacteria. And you have to understand that if we aligned all the viruses that are literally nanometers in length, if we take all the viruses and we align them, we would have the freaking length of a Milky Way. So bacteria, they are an order of magnitude behind that. Way behind viruses. But bacteria are the majority, and they're everywhere. Think about it. There's at least one, for your mass, there's at least 1 kilo of bacteria within you. Most of your fecal matter is bacteria. It's dead bacteria and fibers. So yeah. We live in a micro world, and when Swamp Thing, let me Swamp Thing bacteria. When Swamp Thing, this one. Copy and paste. Let's paste it. When Swamp Thing went there and said, bacteria divided. For billions of years, the kingdom had more than enough biomass to self-organize in the manner of the plants, the animals, or fungus, but it was a peaceful place. No, absolutely not. Living as a bacteria is constant warfare. The other kingdoms were unaware of its existence. Not either. Plants are absolutely freaking aware, because there's a lot of help between plants and bacteria. There's a lot of things that happen. Also, you would think that the red, being all the red cells, etc., would know what aggresses it, since it is organized in order to fight those things. The other kingdoms were unaware of existence. If their subjects occasionally fell ill, which is attributed only to the whims of nature, there was nothing to see, nothing to fight. When we saw you without a thought, now you get her attention. She's occupied with her battles. But at last, the red intelligent monkeys looked through their tubes and found another world below ordinary perception, a frothing microcosm of tiny creatures colonizing, dividing and redividing, infiltrating. The monkey devised substances to battle these invasions called antibiotics, and for the first time, the peaceful ancient kingdom knew war. Beaten back, suffering tremendous loss, the kingdom awoke itself and began to resist. This is absolutely fucking false. Everything about it is so false that it's basically its own universe. Being a bacteria is war between yourself and between all of the other kingdom. All of the other ones. Bacteria, archaeota, and eukaryota. You think about it, that the red, quote unquote the red, of this invisible, this kingdom, the eukaryota, as I'm going to show you the tree again, because this tree is pretty useful for what we need to show. This tree here, this tree. Animals, ciliates, green plant, fungi, flagellate, microsporidia. You have to think about it. Most of the eukaryota are completely monocellular. Absolutely freaking monocellular. So they are also affected by this war. And the fungi. What do you think penicillin is? It's made by penicillium. Fungi are just as microscopic as that. You can see those things. Those things? Yeah, those would be the fiber of a fungi. But no, oh god, no. Swamp Thing is not made... I mean... This is a kingdom of war. And as soon as antibiotics were developed, scientists informed the doctors that if we were to abuse them... So think about it, it's a freaking 40s. But if we were to abuse them, all the bacteria would be resistant to it. All of them. And as soon as we created antibiotics, well, as soon as we had antibiotics, we have selected bacteria to be resistant to them because there's resistant genes. And the main way what we select the bacteria with, if we want to make like a GMO, you have to collect your bacteria, so you have your interesting gene, and right with the interesting gene, you have a resistance cassette. Well, what in French we call a cassette de résistance. Cassette de résistance. And what it does, it resists to a certain amount of antibiotic and a very certain antibiotic. So when we cultivate the bacteria, all the bacteria that we have then, in the presence of the antibiotic, is the GMO. That we'll be able to harvest to have this GMO colony. And any other bacteria that had the resistance gene. So, you know. But normally, if you work cleanly, it's only the bacteria that interest you. So, yeah. Yeah, not really. It's fairly misunderstood. Also, most of the photosynthesis in the world, bacteria, cyanobacteria, also called blue algae. If you want to see the diversity of shapes for bacteria, well, you have monococcus, diplococcus, streptococcus. It's the way they arrange themselves, preferably, after a while. Sarkinia, when they do cubes. It's very funny, they do cubes, because tetracoccus, they do squares, they do cube, and then staphylococcus, they just do, like grapes. They just arrange like grapes. And then you have the spirals with Vibrio, Spiolum, and Spirochetes, Spirochetes. Vibrio, we have Vibrio difficile, which I think is the one that does cholera. Spirulum would be Lyme's disease. If you want to have cogs, you have Staphylococcus aureus, which is the main resistance prone bacteria. It is a super resistant bacteria. When you have a structure like hyphae that you mostly see with mushrooms. Rectangular shaped, I haven't seen any one of them. Star shaped, pretty cool. Filamentous, I don't think that I have seen any filamentous one, or maybe that's the one for Lyme disease. I don't know. Clostridia, Symbiobacillus, Diplobacillus, Streptobacillus. You know, Strepto because they do a chain. Palisades, I've never seen a palisade one. And Escherichia, well, because you have Escherichia coli, so E. coli, the most well-known bacteria that you have in your guts. And that's about it. That's about it for bacteria. You have to understand that the ribosome and the nucleotides and everything here is supposed to be absolutely mixed. And the plasmids too. They're mixed inside. They are very compact. So if you were to look at it, it would be all orange. It would absolutely be all orange. Because what bacteria would quote-unquote lack in anatomical structures, they do in chemical complexity. Their metabolism is incredible, and their reactivity is incredible too. They can have multiple modes of metabolism, where a eukaryote cell would only die. They can shift the way that they translate their DNA, create new proteins in order to resist to another environment. Because think about it. You are a human cell. You are a red blood cell. What you do, you are in the blood, 36 degrees Celsius. The acidity of the blood is completely regulated. You don't have to do a lot. You are a muscle cell. 37 degrees Celsius. It's regulated. Acid is regulated. Oxygen amount is regulated. Sugar amount regulated. Neuron, the same. 37 degrees Celsius. Sugar amount regulated. Temperature regulated. Acidity regulated. You are a bacteria on an apple. Imagine that you are on an apple. You are a bacteria. It is 16 degrees outside. The acidity, you're not in a solution, so you don't really have to care. But you have to care about you depleting your water, because you are a bubble of water. So you need to keep the water inside. So you need to have a resistant form for that. And then after that, you get eaten. So in a few seconds, you are in a solution. You are in water technically. This water is extremely acid. You think about a pH of 2 for the stomach. It is extremely concentrated. You are in a nutrient-rich environment, even though it's very acid. And it is 36 degrees Celsius. 37. It is a complete change of environment. You do that to an animal cell, it's going to die. You do that to a bacteria, it's going to adapt. That's what it does. Bacteria are made to adapt, and this is why it has a wall, a capsule, a cell wall, and then you have the plasma membrane. The plasma membrane being all the cool things that you have, all the cool lipids. The cell walls and the capsules are usually made of proteins, sugar, and special fats. But mostly proteins and sugar. This is a lattice. This is a very important lattice. That changes between gram plus and gram minus. Let me show you the difference between gram plus and gram minus. I don't know why I didn't think about that. But I didn't want to go too much in in length. I think that's good. I think that this one is good. Yeah, I'm going to show it like that. Unfortunately, it's in French, but it's resources nonetheless. Here you have this. You have gram minus. So you have a sugar. You have basically an outer membrane, this is fat. Then you have sugars, and you have a chain of sugars that are extremely resistant. And then you have your plasma membrane and the proteins inside the plasma membrane. And then you have proteins to interact with the environment. Porins. So they let stuff in. If you are gram plus, you basically just have the peptidoglycan, so it's just a chain of sugars. Then you have an interplasmic space, so you have a void. And then you have your lipids of your plasma membrane. And all of that is made to resist to the environment, because you are at the mercy on the environment. You are outside. It can rain. It can be dry. It can be acid. It can be basic. It can be hot. It can be cold. And your role out there is to find water and sugar and reproduce. And you need to resist to all of these weird conditions. So that's what they do. That's what they do. That's what they are made of. That's why they are very complex, and that's why they are extremely successful. Because as soon as you find an environment, a bacteria is going to evolve in order to fit this niche. They are very... The basic anatomy is made for that. So evolving towards that is way easier than, for example, a eukaryote. And there's a thought experiment that if you let, for example, E. coli that divides every 8 minutes, basically every 8 minutes you double the population. If you were to not control if you were to have an infinite amount of sugar, of water, of protein, and of fat to give it, in 24 hours you will have a mass of the Earth made entirely out of this one E. coli that just reproduced. The mass of the Earth. Think about it. In 24 hours. This is made for exponential growth. For resistance. And this is why I don't like that they are underestimated. Because even though only microbiologists Chad are really interested in them, they are some of the most interesting organisms that there is out there. And then you have archaeas. The main thing that changes with archaeas is an extramophile life form that can survive as in any environment. No. An extramophile is basically an organism that you find in a condition that you would consider extreme for human survival. So an organism that is, let's say, that can only... well, it's good that you talk about it, because archaeas are most often considered extremophiles. Most of them are well known for their extremophiles clades. Basically, archaeas, for example, some of them are thermophiles. So they only live at around 80 degrees Celsius. You get them hotter, like about 100 degrees Celsius. You know, 80 degrees Celsius to 100 degrees Celsius. You get them any colder, they die. You get them any hotter, they die. You have bacteria that only grow in very concentrated salt amounts. Like a lot of salt. Or very acidic. Or they can only grow with a lot of pressure. There is a lot of adaptation, very specific adaptations, that enter into one specific environment, which make this thing extremophile. For example, a lot of salt. The Dead Sea life forms are entirely comprised of extremophiles. And they are all adapted, viruses included, even though they are not life forms, to resist this amount of ionic forces exerted by the salt. In the vents, the thermal vents, you have thermophiles. In the Arctic, you have cryophiles that only live at around 2 degrees Celsius, 0 degrees Celsius, sometimes less than that. Hypercryophiles or hyperthermophiles, which are really in the extreme. Problem is, you have adaptation. For example, in the genome, I didn't want to get too much into depth, but I will have some repeats. That's not too bad. Basically, the genome. Let me tell you about something. Let me tell you about it. CNG in DNA is extreme. It's much more stable than ANT. The ANT links. Why? Because CNG are able to link 3 times through hydrogen bonds, and ANT only 2 times. So if you have three bonds rather than 2, it is more stable. And you see that hyperthermophiles or thermophiles, so organisms that lives in very hot environments, basically they have a higher percentage of CG inside of their genome. They have a higher percentage of guanosine or cytosine because it's better to survive in high temperatures. Such is also the case with the proteins. They have proteins that are more able to survive in the hot. Problem is, here's the thing: if you are adapted to survive in hot environments, if you are colder, well, your very, very, very stable bond will not be able to be destabilized if you want to reproduce, or if you want to nourish yourself, or any other interactions. So you're going to die, because you're not going to be able to do any chemical reactions that are necessary for your metabolism. Cryophiles in the reverse order, have to be as unstable as possible, because the cold stabilizes them. The cold stabilizes them, so they have adaptations to survive. For example, water crystals, to do some antifreeze. They have more sugar content or salt content to make the water inside of them less able to freeze, less prone to freeze. And they also have, according to the logic, more ANT. Even though I don't like the fact that they have more ANT, because gene-rich regions in the DNA of stuff usually has a higher percentage in CG. But they will be trying to be more unstable, because the cold stabilizes them, and we are trying to go into how to say, something that is comfortable. So if you put them in something that is let's say 30 degrees Celsius, 30 degrees Celsius is basically, for a thermophile, it's freezing temperature. They die. For a cryophile, it is basically boiling temperatures. They explode. Same with salt content. Salt can dehydrate a cell immediately if you have too much salt. If you don't have enough, the cell explodes. So think about it. If you are not in these very precise ranges of chemical and physical parameters, physical-chemical parameters, your cell, so your life form, is going to die, because it's simply not adapted to this environment. It's like this one picture of the country balls temperature. Australia, I think it's Australia. Australia. Yeah, it's this one. It's this one. It's perfectly this one. It's this one. I need to copy the image, and I need to put it there. So you don't really have something that is able to adapt to any environment, because everything has limits. Everything has limits. But for example, you have here, you have it. You will call here a cryophile. This is a mesophile, so something that is about us. And then you have a thermophile. So 40 degrees Celsius, pretty good. Pretty good. But then you have some problems. It's still comfortable, but it's not optimal. And when it dies, it pretty much dies. There, not optimal, but you can live. You can pretty much live. Here, you begin to have some problems. Here, it's absolutely not comfortable, but it's alright. Then at 10 degrees, it's not life. It would be like 36 degrees for us to have the bacteria that live with us. And for us, 36 degrees. For our cells, this is the case. And then you have thermophiles. They don't like it. They are here. And then you have cryophiles, and they're here. And this is this. Extremophiles are just extremes compared to us. Which is, stuff that happens. I don't think that tardigrades are extremophiles for that, for that really specific reason. Because they don't live in these extremes of temperatures and or pressure and or radiation. They use a resistant form. They basically dehydrate themselves and become resistant until the conditions are favorable enough, and then they become then they relieve. Basically, they come back to life. So yeah, it's also like the frog that freezes itself, basically destroys its own metabolism to basically come back to life once resuscitates once the spring comes. It's not really a extremophile behavior. And then you have archaeota, which basically are just between eukaryote and prokaryotes in terms of chemical composition. They're weird. We don't talk about them a lot. They're not very studied. They're difficult to study too, because a lot of them really like very hot temperatures. So keeping them is pretty hard. There's no disease from them. So they don't really interest a lot of the investors. There's not that much research, unfortunately. But there's a lot. There's some research, but not that much. Not compared to that, and especially to that. Eukaryota. Eukaryota, which is us. Eukaryote, which is us. This is a human cell. This is pretty much an animal cell. And this is an apicomplexa, which is a protist. It is a single-celled eukaryote. And this is a dinoflagellate, which I think you can. It's a eukaryote, so it's a single-cell organism that has silicium. It uses silicon in order to create a skeleton, an outside skeleton for itself, which is glass. It is organic glass, and they are very helpful when it comes to fossil records, because we can identify the silicon inside of them, and then, well, we can find them. Each time that we can find them, we can also find what they are. We can also, basically, if you find that in rock, you know that there was life. That's about it. But yeah, eukaryote are defined by the fact that they have a nucleus in which there is all the genetic information, all the DNA, all of the DNA. The RNA has to pass through there. It has a double envelope here for the. It has a double envelope, and it has. I mean, I don't like the fact that it's seen like that, but it does have a membrane that is made of this stuff. A double membrane. It has a double membrane right there that allows water to be kept inside and be kept out. It has some pores. I don't like how the pores are represented. I much prefer the way they were represented here, for example. It's much more exact, because this implies that everything can go in and out, without any energy that is expended. And it's not the majority. We do have something that we call aquaporin, so aqua for water and pore for, well, pores, so holes, that allows us to have a transfer of water. But we have a lot more transfer of ions and sugar and all of that stuff that needs energy in order to take in the interesting stuff in and keep it in. To create, basically, there's a gradient here and there. There's more stuff in that is interesting for the cell than there is outside. Normally, if that was a passive pore, well, with the laws of physics, it would passively go out and equilibrate the stuff, which happens when we die. But no, a living cell has more stuff in interesting for the cell than there is outside as concentration. It also has mitochondria, the powerhouse of the cell, which has bacterial origin. All of the eukaryotic cells are aerobic. They breathe oxygen. Bacteria are not confined by this notion. Bacteria and archaeas are not confined by this notion, and sometimes oxygen. Yeah, it's for more casual audiences. But it is pretty good. It is pretty cool because it's very complete. Because you can find the centriole, you can find the centrosome. The centrosome is basically the center of the cell skeleton. You find the peroxisome, which is just a crystal. It's a crystal. You have the Golgi apparatus, which is very important for maturation of proteins, and also get stuff from inside to outside and outside to inside. Depends. It's a double way. You have the rough endoplasmic reticulum, which is keep calcium in, also keep protein in, can do some maturation of protein, pretty cool. The mitochondria has its own genetic code, it has its own genes, it has its own DNA. There's some stuff that is kept here, so mitochondria cannot be independent. Lysosomes. What are lysosomes? Lysosomes are just the membrane becomes a pouch, so it gets into itself, and it's a digestion pouch. It's the way that the cell digests stuff. It has a lot more acid inside than it has outside, and that's about it. It creates acid in order to digest stuff, which is sometimes used in immunity. Because we talk about phagosome, phagocytosis. So phagocytosis, so a movement of the cell in order to eat something. It's just a giant lysosome. It's just a big pump, and it pumps hydrogen inside, and hydrogen protons being a factor of acidity. Well, you become more acid inside than outside. It breaks down stuff, and you have some enzymes too that do that. And a plant cell is different. Plant cells are different because they have something else that happens. You know that we have the mitochondria, which is the powerhouse of the cell. Well, plant cells have chloroplasts. And the chloroplast is the powerhouse of the plant cell. Well, the other one. Because rather than do respiration, they do chlorosynthesis. So they use light, and with water and carbon dioxide, they create sugar and dioxygen. And with this sugar and dioxygen, they do breathe. They absolutely do breathe. Plants do breathe oxygen. And they're basically arranged the same, except that they have vacuole, which is basically a patch of water inside, which can take sometimes up to 90% of the cell volume. So you have everything that is completely on the side, because you have a big water bubble inside. And they also have the cell wall, which is made, guess of what? Of cellulose. The stuff we make paper out of. And that is how a plant cell well lives. It is considered biconta, which if you want to have more information, it is considered biconta. And basically a plant cell is just a cell with a wall and a chloroplast. The chloroplast happens, there is a hypothesis, mitochondria happens through something that we call endosymbiosis. So endo inside, symbiosis, so for symbiote, you know, so something that helps. So it's hypothesized that there was, what does the mitochondria do in a plant cell? It breathes. It breathes. Remember, the only stuff that is important for the let's say the race of life is to make more of that, more ATP. We create more ATP through respiration. So using ADP or AMP, depends. Using ADP and oxygen and water, we are able to have more energy that goes through a proton pump, that goes and allow us to assemble ATP, which is the usable energy source of a cell. So basically what that means is that a plant cell only needs water, carbon dioxide and light. It makes its own sugar. So with inorganic compounds, without any organic compounds, it can create, through mineral compounds, it can create its own organic matter and its own energy. It is insane. It is absolutely insane. It gives you so much more energy, and this is why plants are so energy rich when you eat them. They have such complexity when it comes to their they have a possibility to create very, 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 very complex molecules, but we cannot produce, which is why we eat them. Which is why we eat them. And this is why plants are the true ruler of the world when it comes to how to say complex eukaryotic cells. No, not multicellular eukaryotic life. You have dioxygen, you have water, you have so carbon dioxide, sometimes dioxygen, because you do need the production. You do need the production for ATP. And this is why when they are inside of a seed, they don't have any they have a reserve. You know, a seed contains something that we call the endosperm, which is just fat and sugar. It's basically just that, fat, sugar and protein, which is used in order, because the baby plant only uses the mitochondria. And then when it emerges from the soil, it is able to use the chloroplast, and then it can grow. And this is why plants have mitochondria still. But what's thought of is that eukaryotic cells, proto-eukaryotic cells, having already a nucleus through means that are theorized through other means, eat or get a parasite that can absorb dioxygen, which is a horrible. Oxygen is a terrible poison, but if you know how to detoxify, if you know how to make it not a poison anymore, you can make it into energy. A lot of energy, a lot of ATP, which makes you extremely competitive. So it turns out that if you have these parasites that can detoxify the oxygen, you can go where there's oxygen. So you can explore a bit and find more organic compound and maybe eat those pesky bacteria that create oxygen using only using only carbon dioxide and water and lights, using freaking quantum physics, those freaking nerds. And then it becomes a symbiosis and the mitochondria. No, it doesn't become opposite at all. It doesn't become opposite at all because you still need to create the ATP and you still need to breathe. You still need to breathe. The chloroplast is able to create organic compound that is then used to do exactly the stuff that this one does, and even more. So basically, you have an excess of organic matter that you create yourself, and you don't need to hunt. You don't need to hunt for that. So think about it. You are let's go into the example of a single cell. If you are a single cell, how do you get organic matter? Because you cannot take carbon dioxide from the stuff. You need oxygen. You need oxygen, so you have to breathe sometimes. Maybe not. But you also need organic matter. You cannot create it from nothing. Well, what are you going to do? You're going to eat other living beings. You're going to have to hunt. You're going to have to move. You're going to have to use a lot of energy for that. So to get all energy and your own organic matter, because you also have to reproduce, you're going to have to hunt. You're going to have to expend a lot of energy. What plants do is that they say, okay, I don't need to move. I don't need to move anymore. I can just create an excess with stuff that is absolutely everywhere, and that you breathe out carbon dioxide, light, and water. And remember, there's no life without any water. So basically, it's like a cow. A cow can eat the ground. The cell just eats the sky and the water. The cell just eats the water. By eating the water and the gas inside of it, if we take a single cell, well, it has an excess of sugar, but it doesn't have to hunt for it. It has an excess of fat, but it doesn't have to hunt for. That it can use to create its own energy and its own organic matter, and can reproduce very fast. So it doesn't become obsolete at all. It is absolutely integral to that. It's just that you have another part of the system that has been added. That is a cyanobacteria. Because the chloroplast is made of a lot of structure, very complex structure that you find in cyanobacteria. Who needs legs when you have to? Exactly! Who needs legs? And you see how big plants can become? You see how big plants can become? There are the first one that colonized the ground, with bacteria, of course. They colonized the ground. They are the first, let's say, quote-unquote complex life. I would say anatomically complex life, big life. I would say big life that colonized the ground. They are the first one that colonized islands. Moss, when you think about them. They don't need that much more. Remember the game of life that we will do when we will do the evolution episode. It's a game of good enough. So when you have this symbiosis with a cyanobacteria, and this X parasite that you actually have, this X parasite that you domesticated, and this one that you ate, but finally you don't need to digest because you're so small that you're transparent. All the color in the plant cell comes from that. It comes from that. You just eat the red and blue radiation from the light that is absolutely everywhere. Hey! Who needs legs? Who needs to hunt? Who needs to starve? I can reproduce as much as I want. And that was all. Unfortunately, that was all. Unfortunately, that was all for today. But if you have any more questions, I would be very, 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 very happy to answer. I'm just going to drink a bit more water. Because I think that it was just an introduction, but you can see how heavy it can become already. We talked about a lot. We talked about a lot when it comes to life. We talked about a whole lot. And for once it was prepared. For once it was absolutely prepared. But this is the basic stuff that you need to understand in order to have a basic understanding of life. And the way that it's made in order to be a good parasite. In order to be a good parasite. This is the very surface that we have touched. The very surface, but it's going to be much more interesting after that. I mean, I don't know. I don't know if it's interesting for you or me. It's nerd stuff. For me, it's very interesting. For me, it's my passion, my dude. So, if you have any more questions, I would absolutely love to have them. Absolutely love to have them. I absolutely love to have them. Ah, no, I'm cut in half. Ah, no, your immersion is broken. Ah. Oh well. Sure. Let's go. Just to understand, an extremophile is an organism that can survive comfortably in an environment that we can't. Yeah, but just that. We just said, holy shit, this is pretty extreme. Well, this is an extremophile then. Well, shit ass, 36 degrees Celsius is freezing for them. Or maybe our concentration of salt is not enough salt for them. You know, it's just relative to us, they're pretty extreme. Relative to us. That's the whole deal of extremophiles. Relative to us, we're pretty extreme, but if we want to talk about ourselves, we're pretty extreme too. We are pretty extremely, like, quote unquote, in the norm. But 30 degrees Celsius, 37 to 36 to 38 degrees Celsius, yeah, it's pretty specific. It's pretty extreme. It can be even freezing or burning for some. And yeah, maybe I will do an episode on the second episode will be on trophies, which is what do I eat and what do I become through that. Tardigrades are the first thing that comes to mind when people. Yeah, they do, but tardigrades are not according to the definition that I know. Like, let me ask you, because I am not. I want everyone to understand. I want everyone to understand that you have to put into question every single one of my words. I am not a professional. It's been a while that I'm in this world, and I have opinions, so I am biased. If you need to have multiple sources. I give you an introduction. Unfortunately, as a lot of vulgarization, it has been passed through my biases and my opinions and all that, meaning that I can't really be an objective source. And most of my opinions, I'm sure, are garbage when you ask a true professional. But I want everyone to have a quick understanding so that they can have basis, and then they can think for themselves about the stuff. But yeah, they are said extremophiles, but I don't like the term extremophiles when it's used that way. Because what they do is not that they live at 100 degrees Celsius always, everywhere, always. anytime. No. When the temperature is too hot, they go into cryptobiosis. So they dehydrate. It's like saying that the bacillus that you find in milk is an extremophile because it can do spores, and those spores resist to 80 degrees Celsius that we use for 140 degrees Celsius that we use for ultra high temperatures sterilization. Yeah, but resistance forms do not exactly have a metabolism. Like, you can find things that necessitates or live in pretty good like they say that they're very resistant to radiations. Once again they are not active when it happens. Like in the vacuum of space, they're not active. What we're counting is basically we put tardigrades in the vacuum of space. They need to dehydrate themselves as much as possible, as quickly as possible, in order to not explode in the vacuum of space, to resist with radiation and the fact that there's no atmospheric pressure, and the fact that you cannot do drier an environment for literally what we call water bears. You cannot do drier an environment than the freaking vacuum of space. However, there was not a lot of survivor at first, and second of all, they were not active. Once again, they were not active in space. They were in cryptobiosis. So of course, if I put plant seeds, are those cryophiles because they resist the winter? I don't think so. I don't think so. Are those extremophiles because the winter is necessary for the germination of some plants? I don't think so. I think that's an abuse. That's a language abuse, and I cannot control language. I absolutely cannot control language. If I would, I could. Like any self-absorbed person, if I would, I could. But yeah. For me, tardigrades are not extremophiles. And maybe that's a hot take. Maybe it isn't. I don't know. But they're not extremophiles, given the list that I'm going to give you in the next week. Any more questions? Because I do like to answer those. I do like to answer, and I hope that you're not bored. I hope that I'm not boring anyone. Oh yeah, supercut. Oh, supercut can be good. Yeah. Yeah. Extremophiles are the ones that live in extreme environment, not survive. Yeah, pretty much. They necessitate it. Well, yeah, they necessitate it. Thank you. Thankies. I don't think I have any more questions for now. I'll go watch the part I missed. Okay, no problem. No problem. Thank you. Thank you very, very much for having watched this stream, which basically was a year coming. It was a year coming. And I have to say, I was very, very stressed about how it would be taken and how it would go. And of course, there was you and there was everyone else, and I think that was worth it. Finally, I'm doing something that I was supposed to do since the very beginning of this channel. After one whole year. After one whole year. Scientific vulgarization. Finally. Speaking so much, make my voice a bit coarse. I will say thank you so much for watching. It's really important. It's really important for me, and I hope that I will find my niche there. Making shorts out of the stream, and making a supercut. Yeah, making a highlights. Yeah. I will make a hypercut. with some memes and maybe better music. I don't know. But yeah, I don't have any possibility to raid anyone, unfortunately. I don't think that I can. But yeah, thank you so much. Maybe I'll finally find my niche on YouTube. But tomorrow, again on YouTube, will be Blasphemous 2, because I love my smart blasphemia. And I say, have a good day, have a great day, because this was okay. This was a proof of concept. This means that I can do it, and I can do a duration of a normal stream. So that's pretty good. That is actually pretty good. Yeah, and it does attract some attention. So yeah. Thank you. I would love to be able to blink, but no, to you know, do a wink, but I can't. That is a final goodbye for today. That was a short stream. But I think it was very dense in information. Let's go. Have a great day. Mwah. Is the stress gone? A bit, yeah. As soon as I hit go to direct, the stress was transformed into energy, because that's an exercise. Yeah. I think that I can improve the formula too. Which would be nice. Would be very nice. But yeah. See you tomorrow. If you're there tomorrow, it's Blasphemous 2. It's not that interesting. Let's go. Have a great day.